Okay, so what I'm going to do today is kind of an overview of what we're going to be talking about over the next year. And again, I need feedback in terms of if there are particular things, if you've looked at the syllabus and there are important things that even you wanted to talk about that aren't on the syllabus, certainly let us know. We've got opportunities for juggling. I am trying to gauge in terms of, we had thought about doing some small group sessions, and I've got to figure out a little bit in terms of how many people would actually participate in things like trying to learn about your own genome or how to interpret a genome. So we may end up sending you a brief survey monkey to be able to figure out both the level of interest as well as to how many TAs essentially we might need to do those exercises. So watch for those from Kelly in your inbox. So I think one of the most important and first questions which have befuddled us, I have to say, even as we've been thinking about this as a planning committee, many of you know that we've been planning an initiative at the University in Precision Medicine, is what in the world are we actually talking about? What is precision medicine? What goes in under that umbrella? What is it that we're talking about? So I will tell you, this is my definition. It's only my definition. There are other people that would either add to this or take away from this. This is not meant to be the party line for the university in terms of how we're thinking about this. But to me, this is patient-centered, and so it's about providing the right treatment for the right patient at the right time. And to me, it's also about being, again, coming to the patient on their terms, so engaging the patient within that discussion, hopefully with the goal of being able to treat them better, but also to be able to prevent problems and improve their quality of life for themselves as well as for their family. And again, I'm a geneticist. Many of you know me as a geneticist or a genomicist, but I want to be very clear, this is not solely about genetics. I think I'm going to tell you about many of the things that are related to genetics, but it's also about biomarkers, whether they be biomarkers that come from pathology, whether they be biomarkers in terms of serum or protein biomarkers, imaging biomarkers. There are other things that can tell us and predict for us about diseases that might be coming down the road. But it's a lot about risk stratification, a lot about trying to determine for that patient in real time, right there, right now, what do they need to be worried about so they can hopefully plan better for the future, again, to maintain their health, or if they're in a state where they already have disease, to be able to figure out how better to treat that disease. So today what we're going to talk about is our current understanding, specifically from a genetic point of view, of what we don't know and what we don't know, to be frank, in terms of human health and disease. We're going to talk a little bit about, from my point of view, why it is we care about genetic diagnoses, and some of us may, and some of us may care more about it more and less. Also to understand what our current state is in terms of predictive medicine. So I'm going to be making this dichotomy in terms of individuals who are already sick or already have symptoms, and thinking of that in a diagnostic mode, trying to understand the question of why they have those symptoms, versus individuals who are asymptomatic or pre-symptomatic and trying to predict why they might be at risk for these things in the future. So I'm going to draw those two different distinctions, and some people call it slightly different things, but at least I'll present it in that way. And then as we're thinking about this, I also, and I'll weave it throughout all of the discussion that we have today, thinking about not just how this affects a patient, not just how this affects me as a provider, for instance, but also how it affects lots of other people in society, because it's really a lot of the barriers we've seen in terms of implementation had nothing to do with the medicine. It had to do with things outside of the scope of medicine, some of which we've tried to deal with. So the first thing I want to say is that there's a hype and there's a hope and then there's reality, right? And so I want to be very much grounded in reality as we're doing this, because I think and I understand a lot of us want to promise things to patients for the future. Some of us try raising money from donors, and we make this out to be the best, most fantastic thing. But we really have to be honest with ourselves in terms of what we can and we can't do. And so this is, to me, just telling. This is a cover of Time magazine, and it's one of many sort of things we see on the cover of the New York Times, the Wall Street Journal, online, whatever mode you want to look at. But importantly, to realize this came from 1994, right? So this was 20 years ago when people started thinking about the future of genetics and genomics is now. This was something we were supposed to be doing today, and that was 20 years ago. The good news is, since then, we've actually finished sequencing the human genome, the first draft, the second draft, even the third draft, so we're getting farther along, and we're understanding better what those 20,000 genes do. But I also want to be realistic, and I think we owe that to our patients, to be realistic about what we can and we can't do still about this. We still don't know what all of these genes do. We still don't know all of the genes that are involved in human disease. We still, for many diseases, can explain maybe 10% of patients that have a genetic predisposition, but depending on the disease, there's a lot that we don't know at this point. And genetics, unfortunately, is something that we oftentimes know what experiments we'd like to be able to perform to answer those questions, but oftentimes we're restrained. Budgetary constraints from being able to do and answer the questions we'd like to be able to do. But I'll talk about that a little bit more. So when many of us think about this, at least from genetics, we tend to think about things that are genetic. that are very strongly genetic with single powerful genes and mutations that have great impact. And then there are other things that are genetic in the sense that they have genes that contribute, but it may not be all about one single gene or one single variant that's doing that. And so when you look across this space, and if you look at these different quadrants we're talking about, there are many diseases that are quite common, but if we look at, for instance, what we would call the penetrance or how much a particular variant confers disease, many of the diseases that you and I worry about are in these things where they're very common. We could be incredibly impactful if we could figure these out, but we don't really know truly about a lot of the genetic variations that confer risk of asthma or type 2 diabetes or heart disease in terms of myocardial infarctions or breast cancer. There's a lot that we don't know within that space. And this is where we get into trouble with companies like 23andMe, for instance, where they try and say that we're going to be able to genotype you for a panel of single nucleotide polymorphisms or SNPs and be able to come up with these very accurate predictions in terms of your risk of developing breast cancer or prostate cancer or asthma. And it becomes problematic because for many of those risk prediction models, we're talking about accounting for something like 5 to 10% of the variants that we see within those diseases. And 5 to 10% in terms of being able to ascribe for that does not give us very good predictive value. And so if you look at things like receiver operator curves for those predictions, they're completely useless. They really give us just as much information as we would get by our usual clinical assessments. So that's been a problem. And people, as you know, have done these things called genome-wide association studies to try and understand that better. We will surely get better with this over time, but in terms of the overpromising, that's one of the things that if you try and overpromise in terms of offering a 23andMe type of answer right now, we cannot deliver on that realistically and accurately for our patients. On the other hand, and this is simply one moment in time, when we look at this quadrant over here where we're thinking about conditions that are rare or very rare or ultra rare, but on the other hand have variants that are very, very impactful. So in other words, very severe mutations, unfortunately sometimes very rare and difficult to find, those types of things we're now getting very, very good at being able to identify. So any of those things that we call monogenic or single gene disorders, essentially we are almost getting to the point, if it truly is a single gene disorder, we have very, very good ability to be able to figure that out, even if it's a gene that's not yet known. It will get better over time, but certainly we are much better at that quadrant up here than we are at this quadrant down here. Increasingly, we're going to be able to seep in from this quadrant, going farther and farther down to be able to identify rare variants, but for common diseases. And again, I think those are going to be things that are going to have intermediate penetrance as we go through, and I think we're likely to have the greatest success in that after we get the Mendelian disorders sort of accounted for. So within this, the thing that's confusing very much for patients is when you talk about genetics, the assumption is from a lot of, again, layperson's point of view, a gene is a gene. Genetics are genetics. This distinction between what quadrant you're in or how penetrant any of these variations are, it doesn't really make sense to a lot of lay people. So they hear genetic, and a lot of people, if they're sort of at the mindset that they're genetic determinists or that it's all in the genes, they will hear that something's genetic and think that they're doomed. That's just sort of the mindset that they go in with. What I want to disillusion them of the fact, though, and we do a lot to try and educate them about this, is that really a lot of this for the common diseases is about a genetic predisposition. It's about setting sort of potential for the future, but with wide confidence intervals. And those wide confidence intervals are based on the fact that we don't know how people are going to lead their lives, what they're going to be exposed to, developmentally what's going to happen, epigenetic factors that might change over the course of their lifetime. And so you can predict with some certainty, but with very, very wide confidence intervals. And increasingly, I think our hope with precision medicine is that as we're doing this in real time, and as we get evidence not just from the germline from this, but with real-time biomarkers, you'll be able to now come up with greater predictions to narrow that confidence interval and come up with better predictions in real time, not just about a lifetime risk, but something like, for instance, what is a 5-year risk? What is your risk of breast cancer in the next 5 years, not necessarily just what is your risk of breast cancer over the course of a lifetime. And by being more precise and giving more real-time data, it can actually be news you can use. In other words, information that's much more impactful on patients as they think about near-term decisions they're going to make with that information. On the other hand, there's certainly still, and this is especially for those of you who are pediatricians or obstetricians, there's still a lot of conditions, though, where it largely really is genes. In fact, the confidence interval is much tighter within those. We are going to be able to predict with great accuracy some of the most devastating mutations because they just very reproducibly totally destroy the function of those genes and have a very reproducible phenotype. But it is certainly something that's emerging, and we're clearly still defining the phenotype associated with many of these genes and many of these diseases. So this is the hopeful part with me, that although we're born with a genetic endowment, it really are decisions, conscious decisions we make about what we're exposed to, how we lead our lives over the course of our lifetime, that ultimately determine whether or not we're going to stay healthy or in fact develop signs or symptoms of these conditions. And again, this is the motivating factor. By being able to give people enough information so that they can make good decisions for themselves about what types of health behaviors are basically worth their investment. And increasingly, at least what we've observed in terms of behavioral science, is many individuals, if they're not completely genetic determinist, knowing that they're at risk for certain conditions and being able to see that in A's, T's, C's, and G's is a highly motivating factor in terms of modifying health behavior. I'm not saying health behavior is easy to change, it never is, but certainly that's something that's more motivating in terms of what we do. So many of you may be asking yourselves, well, for condition X, Y, or Z that I think about, that I practice, or that I take care of, or that might even be in my family, which of those things are actually genetic? And so this is a graph to be able to, or a picture to be able to show you about the heritability. So of the different conditions that we think about, which ones are really extremely genetic? You know, really, it doesn't mean it's a single gene, but which things are largely due to genes. And so there might be some things that you might find surprising. Conditions like psoriasis, major depression, schizophrenia, even if you didn't think of those, because I don't necessarily think of those as being single genes, they actually have very high heritability. On the other hand, other conditions, and this probably makes sense for many of you, might have to do with, for instance, multiple sclerosis, might have to do with infectious pathogens you're associated with or exposed to over the course of your lifetime, other things that might rev up your immune system for one reason or another. But again, you might be predisposed, but ultimately not go on to develop those conditions unless you're in just the right place at just the right time. Importantly, though, and I want to make a point of this, and this is something that in our Center for Excellence of ELSI research here we focus on a lot about, is things that have to do with the brain and behavior, to me, are especially interesting, because I don't know in all cases where we make that definitional boundary between what's health and what's disease. I think if you were to look at many of us and take personality traits, many of us have a lot of OCD features. That's what got us to where we are in terms of being very careful and being very detail-oriented. I think most of us, I hope, don't actually have a clinical DSM diagnosis of being OCD, but on the other hand, there could be a fine line between that. So where's the definition in terms of where's health and where's disease? In the same way, there are other behaviors that we look at that at least under certain circumstances could be advantageous, but other circumstances could actually be real problems. And so one of the interesting things is, if you look at something like intelligence quotient, there certainly is quite a bit of heritability that goes into IQ. I'm not saying clearly IQ is 100% genetically determined. There's a lot clearly that we do in terms of educating young people to be able to help them with this, but behaviors that we're seeing, things like schizophrenia, IQ, depression, all of these things, I would say are not necessarily just dichotomous traits either. To a certain extent, they're quantitative traits, and they're traits that obviously change over time. And so I think that's one of the things we also have to be careful about when we're talking to the public, when we're talking to our patients about these, especially when it comes to neurobehavioral and psychological features. So one of the things that I continue to be frustrated, and I'm intentionally putting a shout out because I know someone in this audience is actually going to figure this out, and it will make us all, the rest of us, much happier when we can do this. Our most cost-effective way of being able to use genomic information right now, our easiest, cheapest way of doing this is by family history information. It is incredibly frustrating, I think you realize, and I'm talking for all of us as a group of practitioners, that we don't have an effective way of being able to collect and use the family history, even though it's something we've known about for a long time. Our practice, in terms of Columbia docs, we have not effectively figured out how to use this information. And again, I would put the emphasis on our patients are our partners. They're working together with us. They need to help in terms of being able to collect this information. They sometimes need to work with their families to collect this because they may not know all of this information at any one time. And I would be provocative and say that I would hope that families could actually work together on this. That is, that we have enough in terms of health information technology for families who do want to aggregate data and work together to actually combine and put together a combined family history online. And wouldn't it be wonderful if you could figure out a way of being able to do that, and with a push button, be able to push that into our EHR here at Columbia Docs, push it into Crown, be able to do it in a way that we could see it, and electronically we could run algorithms, for instance, to look at things, to look at, for instance, number of first-degree relatives with a certain diagnosis, age of diagnosis, and then use a risk stratification tool to be able to figure out for our patients sitting sitting in front of us in our primary care office, are they at higher risk of colon cancer, breast cancer, anything else? Wouldn't it be wonderful if that could actually pop out to us, push out to us at a clinical encounter and say, you need to think about, possibly in terms of decision support, you need to think about starting to do screening at an earlier age, or you need to think about enhanced methods of screening because this patient is at higher risk, or this patient might benefit from talking to a genetic counselor or going through genetic testing for more refined risk stratification to see if in fact they carry, for instance, a BRCA mutation if we're talking about breast cancer. So these to me are frustrating because they're solvable problems. We simply haven't, as far as I know, figured out a way to be able to do this. There are other folks that are trying to do this. So there's a consortia, as many of you know, of NIH-funded investigators in the eMERGE network that are trying to figure this out. They still haven't figured this out. This is the Surgeon General's sort of answer to being able to do this is everyone next Thanksgiving to meet with your family, share your family history, be able to put this in a common tool online to do this. There are other commercial companies that are thinking about this, but I really shout this out again to those of you that are in health IT. If you could figure out how to do this and how to commercialize this and how to get Epic to actually take this up into their package, there's a lot of money for you to make on doing this. So I put that out there as a challenge for you. After that, in terms of being able to use that as an inexpensive way of doing this, we have some other ways of being able to be more precise about this. And so this is now coming to the idea of genetic testing. What have we actually got? What's in actual clinical practice? What can individuals access? And then again, I'm going to put a shout out to those of you in health IT. How are we eventually going to integrate that information into the electronic health record? Because right now I can tell you based on where we have it in Crown, it is buried. I know where a lot of it is, but many of my other colleagues don't know where it is. And I can tell you I'm getting tired of the emails that come and say, Wendy, where is this particular test result? How can I get access to this? We need to make this a way that it's actually being integrated, for instance, putting on a problem list, or as the information evolves and we reinterpret information over time, that this is more of an automated system. So one of the things that I ask myself about whenever a patient comes to see me is if a patient is already symptomatic, do they actually want to have a genetic diagnosis specifically? So they're running around with a diagnosis like, for instance, a seizure disorder or breast cancer or whatever it might be, migraine headaches. They're running around with a clinical diagnosis, but do they really want to address and answer the question of fundamentally what's the etiology, what's the cause of that? For many of my patients, and I can tell you some of you have seen patients like this, I've had patients who have been on diagnostic odysseys for more than 15 years. I mean, there's some patients that are really, really driven to find this out, oftentimes because they're very impactful in terms of on themselves and on their families, and there's the hope that knowing what this is will actually effectuate both better information about prognosis as well as ultimately treatment for this. Some of our families, and for those of you that are in, for instance, OBGYN, think of this a lot. A lot of it has to do with risk of recurrence, so thinking about for themselves or for their other family members, will this happen again? And if again, for instance, the problem is a child who's been severely intellectually disabled, autistic, might have a serious birth defect, might have died of this condition, knowing whether or not this is going to happen again has severe impact in terms of whether or not people are willing to even think about potentially having another child with this. Finally, I'll say, and I don't underestimate this, just having an answer to the question of why just puts people's minds at ease. And I know that sounds kind of silly, but just being able to have closure, being able to have an answer, and being able to say that it wasn't something that they did that provoked this is actually incredibly reassuring to them, as well as knowing what this isn't. That is, being able to put a box around it and say, I don't have to worry about this, that, or the other, that I only have to worry about this circumscribed set of things, and be able to focus their attention in that area is incredibly empowering to people. Even though many of these times a diagnosis doesn't provide immediately the treatment, I can't tell you the number of families who become extremely energized about knowing that now they have to fight for this condition here. That is, the molecular target is X, that they can think about how to focus on that particular area, how to work with researchers, how to partner with this, and move the field forward for that defined set of whatever it might be. And so don't underestimate our patients. They actually are very much willing to partner with us in doing this. When it comes to the reproductive options, they really are quite amazing in terms of what we can do at this point. So many of you know about things like amniocentesis and chorionic villus sampling. So once a pregnancy is already established, being able to make that prenatal diagnosis. But again, I want you to realize that we try and think about what is actually appropriate and what the value system is for our patients. And our patients, you realize in New York, we have this wonderful opportunity of working with patients from all over the world with all types of belief systems. And one of the things that we see is that clearly having an abortion, if you were to do that prenatal diagnosis, is either at the very least distasteful, if not really something that morally, ethically, religiously, they or their families would be opposed to. And so we've developed, and again, this is real, this is what we do here at Columbia, down the street at Cornell, all the time, pre-implantation genetic diagnosis. That is, we can make, if you will, test tube babies. We do this, you know, it's not trivial, so the ladies, I think, realize this more than the men do. It's, for you guys, it's really not that complicated, but the ladies have to go through hormone injections to do this and egg retrieval. But to do this, we can actually make a test tube baby and be able to, at the 8-cell stage, or perhaps larger, be able to remove 1 or 2 cells from that and genetically determine which of those test tubes essentially are going to have an embryo that is going to be at risk for or develop those particular conditions. And that, again, is something that, as far as any particular rabbi, priest, clergy that I've met, we've been able to say is acceptable and something that our families have used quite frequently. The issue that comes up, though, is that this is currently not very often, in very rare circumstances, have I found that it's actually paid for by insurance companies. And so this, as an option, is usually about $25,000 per cycle. We can come up with a way of doing this for almost any single gene disorder, but it's not necessarily accessible to everyone. So what does this set up for us in society in terms of haves or have-nots? Can you prune your family tree, but only if you've got enough money to be able to do this, only if you've got a charitable organization that can support you to do this? And at what point do we define disease? So again, as I was talking about before, if there's something that shaves 10 IQ points off of it, some genetic factor, and I know of many such genetic factors where the disability is shaving 10 IQ points off of it, is that disease? Are we ever going to think about enhancement? So not just on the downside, but are we going to think about genetic factors that may actually increase your IQ, may give you certain personality or behavioral traits? Is that going to be okay? Certainly I can tell you right now in terms of IVF and PGD, the most common reason it gets used genetically is actually for gender selection, trying to determine whether you're going to have a boy or a girl and whether or not you want a boy or a girl. I'll say, and I'll make a stance about this myself, unless you've got an X-linked condition, I don't consider it a disability to be either male or female, and so that's not something that I personally endorse in terms of doing that, but certainly it is something that, just so you know, consumers out there, you know, the market is that that's something that people clearly have desire for. So I think part of the thing why we're bringing so many people together to think about this is not just a medical or a technological issue, this is something that as society we have to think about as well. What are the boundaries on this and what are the ramifications? We need to think 3 steps ahead of ourselves as we're coming up with and offering some of these new technologies. This is a slide many of you have seen. This is what's happened with the cost of sequencing over time. Here on the x-axis is time. This is a logarithmic scale with the cost on the y-axis here. You can see that there have been times when there have been great declines in the cost of sequencing. So this is when next generation sequencing came on board. There are going to be further declines in this, and if Illumina would be a little bit more generous in terms of their pricing, the price of sequencing would go, in fact, quite a bit down further from where it is now. But the point is that this has been a remarkable enabler. That is that the technology is now available to at least sequence a genome, and we're getting to the point of being able to sequence sequence an exome and increasingly a genome at a reasonable price. And so just so that you know, again, clinically, you can order an exome sequence. This is something that is readily available. We've been doing it here at Columbia for over 3 years now, so it's something we have great experience with, and we're now getting to the point of being able to clinically sequence a genome. Certainly still some challenges with interpretation, but we're able to do this increasingly well over time. So just as a very brief description for any of you who aren't geneticists or don't think about this, we have various different technologies to inventory our genomic information. We can do it at a very high level in terms of looking at chromosomes, and the analogy here is looking at a set of encyclopedias and being able to do an inventory of this. My 5-year-old son can basically, at least from a counting point of view, count to 46 and figure out if we've got 46, 47, or 45 chromosomes. So this is relatively straightforward to be able to do. But on the other hand, getting the higher resolution is somewhat difficult. And so we've developed technologies including chromosome microarray that don't depend very much on just how good your eyesight was or whether it's the end of the day in terms of the user interface for this. We have now automated ways instead of karyotypes to be able to increase our throughput, increase our quality for doing this. And this can now get to the level of resolution of, again, if it were a set of encyclopedias, seeing a single paragraph that might be missing, a chapter, a page, a paragraph. And so you think in molecular techniques, we can refine the the detail, the precision, the resolution of this to the point that we can get down to single exons if we wanted to. The technology is there to do that. In terms of genome-wide scanning, we tend to have a resolution with a limit of about 100 kb, but we could technologically do this and enrich in certain parts of the genome where we wanted to get as much resolution as we had to. In certain cases, though, it's a single letter, this B that's changed to a Z, a single 1 out of 3 billion base pairs that we're looking for, that needle in a haystack. And even though it might be a very small genetic change, it has profound influence in terms of a gene that might be an important, for instance, regulator, transcription factor, some chromatin modifier, can have profound impact in terms of human health and development. And so no change is too small when we think about impact on human health. The other thing that's somewhat confusing, at least to the lay public, is that not all genetic things are inherited. And so I can't tell you how many times patients come to see me and they say, but Dr. Chung, I have no idea why I'm here. Our child has this particular condition, but we have no family history of autism or no family history of seizures. I don't see why we're seeing a geneticist. And the reason for that is, and it's especially true of pediatric or or prenatal cases, very, very severe disorders, there's a fair frequency of de novo mutations. That is, new mutations that were not inherited from mom or dad. But if you can imagine it and think about it, if you have a mutation that produces a disease that's so severe that people will not live to reproduce, how in the world do you actually propagate those mutations from one generation to the next? And the answer is that you don't. They end up being essentially born each generation and dying each generation, and they arise as de novo mutations. And so we increasingly realize that, especially, as I said, for either fetal or pediatric diseases, a large amount of the disease burden on things that are extremely impactful in terms of health are actually due to this mechanism. The reason this is so important is that these are actually relatively easy to recognize. We now have the ability to actually sequence an exome or a genome of a child with one of these disorders, a mother and a father, and very, very quickly, informatically sift through all of that information and determine for any one child with that condition what's new in that child that the mom or didn't have. Now, I want to disabuse you of the fact. It's not that these are rare events. Every single one of us have these de novo mutations. On average, all of us have approximately 100 of them in our genome, but in terms of the ones that are impactful for actually changing genes, changing function of genes, predicted functions of genes, each of us only have one of those on average. And so being able to find that one needle in the haystack is now relatively trivial to be able to do and determine whether or not that, in fact, is the cause for those children. And that has been, I have to say, huge in terms of understanding diseases that we had no way of gaining insight into before. So again, many of the diseases that were sporadic, where we didn't have families, where children didn't survive for these conditions, we now realize that these are incredibly important within this. So in terms of the different types of tests that we have, one of the things, and this is one of the things we'll be teaching you over the course of this, is we can go everywhere from doing a chromosome analysis to chromosome microarrays to looking at single genes. So for instance, individuals that have Huntington's disease in their family, we can look very, very specifically just at the repeat associated with Huntington's disease, to we can at this point look at entire genome and entire exome within that. There is all of that that's available and everything in between. The limitation of this, though, so all of these things are possible. The cruel reality of all of this is that it's not trivial. And so one of the important things is in terms of cost. It's not just absolute dollars that are required to do this, but the cost to insurance companies or third-party payers for doing this. We are in a dynamic phase right now where we actually have to demonstrate as a community the clinical utility of this information. I can tell you firsthand, insurance companies aren't going to pay $10,000 for these tests if they can't see what it's going to get them. How is it going to improve the health, the life of that particular patient? And in particular, they want data. They want to be rigorous about this. They want to be evidence-based. And at this point, as a community, we have mostly anecdotal data. We don't have large series in terms of being able to demonstrate this in a very rigorous way. And so one of the things we and others are working with in terms of, even if you can imagine it, people from the business school, people from the Department of Economics, people in the School of Public Health, we need all of those types of folks to teach us how to be rigorous about those types of analyses. Another issue is turnaround time. For many of the tests we're talking about, we actually get answers that are within 4 weeks, 8 weeks, 16 weeks even. And you can imagine if you're on the front line trying to take care of a patient, you want results in the matter of a couple hours, a couple days in some cases, certainly within a week. And so one of the challenges that we're dealing with, and we are dealing with it, is how do we actually decrease this turnaround time? It is possible. It's not trivial, but it is possible to be able to do. And so one of the questions we're going to be asking you as clinicians is how important does it matter, and in what clinical circumstances does it really make a difference, either because it's a difference in terms of saving the life of a person, changes in management that we would do, and ultimately improvements in health, as well as reductions in the cost of that. We're in a dynamic phase, as I alluded to, where we know a lot about genetic variations, but we clearly are far from having cataloged all of them. And so I can tell you that if I sequence your exome or your genome at this point, I will come up with hundreds to thousands of genetic variants that the world has never seen before. And understanding exactly what those mean in the context of you and your family sometimes can be quite difficult to do. We have all sorts of computational tools to do this. Some of you, in fact, are the ones that develop those types of algorithms, but we don't have functional data for most of those, and clearly that's a rate-limiting step in terms of what we can do. So how can we get better in terms of the informatics, and more importantly, how can we do better in terms of aggregating that data? We shouldn't be doing this just solely as Columbia, as only Columbia doing this, but how can we really think about aggregating the data across large numbers of other research universities and research enterprises, and being able to all get up to speed and get smarter faster, while at the same time protecting the privacy of our patients and the wishes of our patients in terms of what they signed up for when they signed up for these clinical studies or for these research studies. In addition, and the last of these, which just hits me especially hard, is I can't tell you the number of times we've had an N of 1. That is that in counting this up, as we did earlier this week for a family meeting, there are over 25 new diseases that we actually ourselves have identified right here in our practice here at Columbia, where we have had the first person in the world with that condition. And in a sort of a disheartening way, to be the first person and to realize that you're the first person and that you're on this new journey, this new leg of this journey, is quite sometimes sobering. But on the other hand, as we had a family meeting with our first 10 families with a new disorder, the joy that comes with actually knowing you're not alone when you become an N of 1 to an N of 2 to an N of 10 and ultimately Ns of 100 is actually incredibly empowering. And again, working with our patients, that's the type of thing I think we can catalyze going forward. So I just want to give you a couple examples, concrete examples of how this information in a diagnosis really matters. And some of you may have heard me talk about some of these cases. So this was a patient that was bittersweet for me. It was a 9-year-old who came to me who had had problems with seizures since the time she was born. She also had intellectual disabilities and some movement disorders associated with that. Her family had been searching the globe for a diagnosis, and she finally came to us. She had no family history of any of this, and so it wasn't immediately clear that this was going to be a genetic condition. At the time, we had and still have various different options for genetic testing, everything that go from these mundane things I've told you about, single genes, panels of genes, arrays, exome sequencing to do, and in fact we ended up finding the answer from a panel of genes, which was a relatively efficient and inexpensive way to go in this and very quick. And the bittersweet was that we found out that she had a mutation in a gene for a glucose transporter. So this is a particular condition where she has a mutation in one of the 2 copies of her gene, and her ability to get blood literally across the, or get rather glucose across the blood-brain barrier, getting it into her cells, both her neurons as well as her glia, is in fact relatively compromised. As a result of that, it's as if she's hypoglycemic in her brain all the time. And so as a result of that, she was basically seizing constantly because she was eating a regular sort of diet with this and unfortunately being starved in terms of her brain. The reason it's bittersweet is because this particular condition actually has a ready fix. That is, all I have to do is change her fuel source. Instead of giving her leaded gas, I need to give her unleaded gas. So in other words, what I need to do is switch her from using a fuel source of carbohydrates to now using a fuel source of fatty acids and protein. And by simply doing that and switching that fuel utilization for her brain, and I can tell you this by point of fact, she is now cured of her condition effectively because she's on a ketogenic diet, she gets no glucose, she doesn't rely on glucose whatsoever, and now she is seizure-free and in fact doing much, much better. The bittersweet of this is that she's 9 years old and she has gone through 9 years of epilepsy in which she's basically fried her brain. And so she is unfortunately to a certain point had irreversible brain damage that had I known about this at 9 hours or 9 days of life, I could have had an incredibly different trajectory for her going forward. And so again, as we're doing this, I think what we're trying to get input from and trying to think about this is when do we need to know a diagnosis early? When do we need to know it fast? When does it have profound changes in terms of our health outcomes? And this is certainly meant to be a case in point in which you could have this diagnosis, and if you knew about this when she had her first seizure, not her 9 millionth seizure, you would have a very, very different outcome for her. So it's bittersweet, but clearly something that is still powerful for her, even if it's from this point onwards. This is another patient with breast cancer. I see a lot of women who get breast cancer. For us in the room as women, 1 in 8 of us will actually ultimately develop breast cancer. This was a woman who didn't seem so exciting when I first met her. She had had ductal carcinoma in situ, or sort of a precancerous lesion at the age of 55, which is not particularly young. Her family history wasn't all that credibly interesting. This is actually a picture of her family history, and this is her here. But she had had some other cancers, and that was what made me think a little bit more about her. She'd had a thyroid tumor as well as squamous cancer, skin cancer, but otherwise her family history looked relatively benign. In the old days, before when we had gene patents on BRCA1 and 2, many of you know that we could order BRCA1 and 2 from a company called Myriad, but that was essentially all we could do. There was innovation that was stifled within the community because there was a lockdown in terms of being able to test genes. Those patents have now been taken away by the Supreme Court, and I can tell you there has been no greater change in the area of hereditary breast cancer than opening this up, where we no longer test just 2 genes, we test 12 genes, we test 24 genes, but we are clearly learning a lot more very, very quickly about what is the underlying basis for hereditary breast cancer, not from our research studies, but actually from the front lines of clinical care. So many of the laboratories that are doing this are now starting to hit 10,000, 20,000, 50,000 women, where we now have data now on large numbers of women, on large numbers of gene that have been better risk stratified. These are the genes, again, for BRCA1 and 2. There is very, very good, solid clinical evidence. In fact, this is the strongest clinical evidence we have from third-party payers in terms of what actually impacts care. This is what's responsible for at least the majority of clearly hereditary breast ovarian cancer cases. And for those of you who didn't know, this is Angelina Jolie over here. So Angelina Jolie, for instance, carries a BRCA1 mutation. Her lifetime risk of breast cancer was about 65%. Her lifetime risk of ovarian cancer about 35%. And that's why she made an aggressive decision about getting bilateral prophylactic mastectomies. There has been no single factor that has actually changed public opinion as well as public acceptance of this than Angelina Jolie. And I think that's another important factor in terms of those of you who are in the area of journalism or health communication. It's actually these very concrete stories that large members of the public can relate to and accept and embrace, that Angelina Jolie is still beautiful, she's still gorgeous, she's had beautiful reconstructive surgery, and to accept the fact that that's okay. That's an acceptable solution. On the other side, and I'm not saying this is good or bad, our rate of mastectomies has gone up tremendously after the Angelina Jolie effect as well. So it's very interesting to actually see what influences patients in terms of their decision making as a result of this. So as we do this, and again, this is something that Kathy Crew, for instance, is now doing in clinics, can we come up with ways of being able to find women like this in our primary care setting? So in our ACN practices, in our OB practices, are there ways that we could, again, get patients to use family history tools, individual health risk assessors, to be able to do that and be able to partner with us and be able to risk stratify and say, I'm at a higher level of risk. Should I get that BRCA1 test? Should I get that panel test? And doing that based on these factors that we see. For those of you who happen to be of Jewish ancestry, as I'll show you in a second, 1 in 40 of you carry mutations in BRCA1 or 2. Regardless of your family history, regardless of your gender, regardless of whether or not you've ever had breast cancer, 1 in 40 of you have that type of risk that we talked about. And to me, that's something that actually, and if you do the cost analysis, could be incredibly impactful in terms of identifying those individuals. So these are the spots that we see in BRCA1 and 2 in the Jewish community, here, here, and here. And again, we can actually run this type of test for less than $100 per person. We could actually identify, if you wanted to, from even something as simple as a saliva sample, those of you who might have those types of very high risk for hereditary cancer. I will say I disagree with the idea that everyone in the general population should be getting this screening. On the other hand, like I said, for something that's cost effective like within the Jewish community, I think you can actually make a very strong argument for something like this. So our patient went through, she actually ended up getting a panel of tests in this particular case, the one in the middle, for her hereditary breast cancer. And what was, in fact, surprising to me at the time, for those of you who know this field, was that she actually had a relatively rare type of germline mutation that caused her breast cancer, her DCIS, a mutation in a gene called P53 associated with Li-Fraumeni Syndrome. Many of us think of these oftentimes in association with somatic mutations associated with a wide variety of cancers, but in her case, this is actually something that she was born with. This has big implications for her her in terms of her future risk of other diseases, other cancers in particular. So she's at much, much higher risk, we now know, for things like sarcoma, lymphoma, leukemias, and there now are just emerging data to show that if in fact you're very aggressive about screening, that this can be life-saving by detecting early cancers at an early stage for individuals with Li-Fraumeni, and that we also want to, in these cases, avoid radiation. And so in a case like this, for instance, I wouldn't want to be doing a lumpectomy with breast conservation and radiation. We would want to be more aggressive in terms of surgical options and minimize her exposure to radiation. So as we've done this, we've also, as I said, had an experience for about 3 years now in terms of doing exome sequencing here at Columbia. There have been now a growing number of indications, so we've thought about this in everything from particular individuals who've had blindness, deafness, different types of cancers, autism, intellectual disabilities. We've looked at some cases that are familial cases, others that look like sporadic cases, and we are still now getting more and more evidence. I would say this is not just us, clearly, at Columbia, but increasingly across the country that people are doing this. And as we're doing this, we're starting to understand and appreciate what really is the clinical utility. What are the clinical indications that give us answers? And as importantly, what do people do with this information? What do providers do with it? What do families do with it? What do insurance companies do with it? How does it affect how people think about this? And do they undergo needless testing as a result of this? Could there be a downside? Or potentially, how do they use this in a proactive, positive way? So just to give you a sense of what the reality is in terms of what we can do, as we've done this in our series here, approximately 25% of the time we will find a cause if the indication is a birth defect or developmental delays. If the cause is epilepsy, we'll find the cause in about 15%, and in some cases individuals have had other diagnostic testing before this, so this is what I would say is the lowest possible yield or the most conservative estimate. In familial conditions, we've had in some cases slightly lower estimates if the condition wasn't very well known. But increasingly we're getting more and more data, and importantly what we're realizing is that even in many cases where we didn't make the diagnosis 2 years ago, as the scientific era is changing and as new genes are identified for these diseases, that number actually keeps ticking up over time. So it is not infrequent that I will actually now, on something like a once-a-month basis, one of those undiagnosed conditions suddenly reverts into being a diagnosed condition because a new publication comes out describing a new condition, and we have a small number of variants that we're watching in our patients that we then see, and they come and they hit us when those new publications come out with that. And it's again that sort of iterative process that we need to build into how we analyze and interpret those genomes over time. So for our series overall, we get an answer about a third of the time with this. The challenge that we're dealing with now, and it's an exciting challenge, is many of you know this sort of debate we have, exomes versus genomes. So we're taking many of those cases that were negative on genomes and now trying to look at them or rather negative on exomes, and now trying to look at them and getting more comprehensive information by a genome and seeing if there might have been hidden clues that we would have missed on the first pass. On the other hand, one of the things we clearly are starting to realize is that this sort of, there's a lot of murkiness between what we're doing in terms of clinical care and how that's bleeding into research. And clearly, at an institution like Columbia, the reason why many patients come to us here is because, in fact, we are so good at research. We can go beyond just the standard clinical practice that they can get in the community. But as we start finding these new diseases with an N of 1, what do we do with that? How can we actually go on to prove that what we truly think in our heart of hearts is probably a disease-causing mutation really is? Can we figure out a way of doing functional assays? Can we figure out a pipeline to potentially go from gene discovery to now therapeutics and improvements in health with that? And so I think we've got a lot of opportunities, but we certainly haven't yet realized all of those. In the interest of time, I'll just say very, very briefly, this is one child in one case in which we did this. It was a child that actually had a brother and sister in this particular case, shown here, and they ended up having an unusual constellation of both leukemia in the little girl, as well as what looked like some autoimmune disease, and both of them autoimmune hepatitis in particular, as well as thyroiditis. Ultimately, we ended up showing that there was what we thought was going to be a mutation, and I use that in terms of meaning that it had functional connotation, that this was actually disruptive of that gene in a gene called SH2B3. But just to give you an example, the amount of time and energy it took to be able to prove this was the heroic effort of one of the postdocs here that took almost a year of their blood, sweat, and tears to be able to go through and prove this and prove definitively that this really was the cause of the leukemia by also looking at a large series of other kids that had the same type of leukemia. So again, we're committed to coming up with answers for patients, but it oftentimes takes quite a bit in terms of investment and resources to be able to do that. I'll just breeze through this to be able to say the other thing that's been interesting in this from an ethical point of view, because again, ethics is sort of a common thread throughout this, is we could figure out now that we knew that the problem was one that was essentially limited to the bone marrow, we knew that a cure for this family was going to be a bone marrow transplant. And when we went back to do this, and this has been true of many of our pediatric oncology cases, understanding that and understanding other siblings who could be donors and other siblings who might be at risk for developing the same exact condition is very important to know, so that you don't actually do a bone marrow transplant with another sibling who's just going to have the same condition. As we did this, we realized that none of the SIBs were actually a good HLA match. None of them were 6 out of 6 matches. And so the family, who was very much interested in their own right for having another child, wanted to have another child, wanted the child to be free of this particular condition, because we still don't know exactly what the clinical course of this is going to be. And so they proposed the provocative question, which many of you have probably already thought about in your heads. Can we use preimplantation genetic diagnosis? Can we actually create, essentially, or select an embryo that's going to be free of this condition, but at the same time is actually going to be an HLA match for the kids, so that they can actually become now the bone marrow donor for for these 2 children that are going to be affected. We had very, very long discussions with this, with our ethicists, with our clinicians around this, and ultimately we decided that the family had the right to be able to do that. They were planning on having a child anyway. We have not yet done the bone marrow transplant, but I will tell you that this fetus is now in the 3rd trimester, is going to be born very soon, and in fact we will see how this story goes on to the next chapter. As you do this, and as you do a lot of this interrogation of a genome, there's a lot that you find out. In some cases, a lot that we don't know exactly what it means. But the warning that I will give you that are doing research in this area is think about it now, because you will stumble upon this information, and you need to have a game plan about what you're going to do with that information when you do stumble on it, because it will happen. So as you think about it, I think in this current era, not necessarily in past eras when we didn't know this was going to happen, but prospectively, as we realize this is an opportunity, we need to think about what to do with that information of these secondary findings. And I won't go into this in gory detail today, but again, this is an area where we certainly have had much heated discussion within the ethics community about what you do with this information. And I'll just summarize it by saying that I'm pro-choice. That is, I think patients have the right to have autonomy within this, to make decisions about what's right for them and for their families. And so actually, I'm proud to say that our pro-choice position actually reversed the national guidelines about what was done. And so we now have patients, rather than having results forced to them, actually have patients that make the decision about whether they want this information or not. The vast majority of them decide they do want this type of information that could be life-saving for them and life-altering, but ultimately I think it's important to give those patients the choice about what to do. So now these secondary findings are a defined list of 56 genes. It may change, it may dial up, dial down over time, but at least 56 genes that really do have significant health implications where many of us feel we have a duty to warn. That is, that these are indications that could be indicative of sudden death, where someone could drop suddenly, cancers that could be prevented by screening, and so now families have the ability to get this type of information. And I will tell you that as we sequence these types of exomes or genomes, this happens about 3 to 5% of the time. And that's why I say it will happen to you if in fact you use that type of technology within your research studies. So I think our challenges and what we're trying to figure out going forward for this next year are some of our new opportunities. What are we going to do in terms of cost effectiveness, in terms of demonstrating this? As this information increases over time, how can we make sure that we remain at the cutting edge for being able to do this? How do we improve the diagnostic capability of making our turnaround time faster, making this cheaper, making the interpretation better? And as we do this, though, making sure that this is completely accessible to all of our patients. That is, that this is not something that simply the haves, but the have-nots don't have. And importantly and specifically within the area of cancer, it's not just sequencing the germline, but it's actually sequencing the other genes, which are those of the tumors themselves, either the primary tumor or increasingly also the metastatic tumors as well. So as we're doing this, I think we have incredible opportunities ahead of us. We have the first examples now of true precision medicine. Many of you know about the company called Vertex that has developed this medication, Kalydeco, for cystic fibrosis. It works for about 4% of patients that have one specific mutation in CFTR, G551D. But on the other hand, it is absolutely a blockbuster for those particular individuals. It's, for those of you who have been watching the media, even in this last week, it's posed some interesting questions about what foundations do in terms of partners careers with science, and I will just leave it at that. It'll be one of the topics we'll continue to discuss. I think there are incredible opportunities with spinal muscular atrophy. We're actually getting ready to start a pilot of newborn screening for spinal muscular atrophy to be able to enable some of the actually development of new therapies specifically at a molecular basis for that disease. And I think there are many more examples you're going to see as the course goes on. There are exciting new opportunities in terms of gene editing. We certainly, many of us use this as a research tool to be able to make model organisms, whether they be in vitro or in vivo systems. The day is going to come, I hope, when we actually have the ability to do that in our patients, where we have the ability to literally make a correction of some of those genes. And as we do it, though, we're going to need to be very, very careful that we don't do any harm within that process. So within all of this, those are the things we can do for patients who are already symptomatic. The exciting opportunity I would pose, though, is that we can do this also in a predictive way for patients who are already healthy and keeping them that way. So what can we do in terms of giving people that early warning side? What do they need to pay attention to based on the information within their genes or based on other biomarkers so that we can keep them healthy and keep them engaged within that process? As we do this, we clearly know that none of us is perfect from a genetic point of view. I can tell you myself, I have over a dozen mutations in various different things. Some of these actually affect some things in terms of reproductive issues, and so end up being either a big issue or not an issue, depending on what your partner's genome is like. But other ones very much actually influence health and our predisposition to health. And many of us, I think, wish that we knew some of the information about ourselves a long time ago. On the other hand, not everyone feels that way. And certainly, as we've talked to patients, there are certain patients who would rather be blissfully ignorant until and unless they get to the point where really they starting to have those symptoms, and I respect those decisions. Within this, there are areas of pharmacogenetics that we can think about how to be able to avoid adverse reactions, everything from malignant hyperthermia at one extreme to other things that are much more mundane. But again, the issue is that not every drug, or at least at that dose, is right for every patient, and how can we do better at that by getting people on the right medication, the right therapy that much faster? And I would pose again for you that are in health IT that we need to build decision support into this, that those of us who are busy practitioners, we can't remember all that genomic information for any one patient. Patients clearly don't remember this, but on the other hand, if we can build an EHR, a smart EHR, to tap us on the shoulder at the time when we're getting ready to prescribe that medication and help us to know to avoid certain medications in certain patients or to alter doses, that will be an enabler. That will totally be a game changer in terms of how we use this information. So at this point, the good news is that for many of you that are in this room that are sort of my age and older, the fact that we've survived as long as we have, our genomes are not too bad. Just trust me on that. For those of you that are a little bit younger and those of you that are still having kids, I can't give you the same guarantee. So, but that is certainly one of the lessons that we've learned. And clearly, as I said, many of the mutations, unfortunately, are in children who don't survive to be able to live to be the age we are. And so, again, the utility, I would actually posit, is even greater in the either unborn or the young in terms of this. As we talked about this, I think the great opportunity we have here at Columbia is going to be to build this whole pipeline and thinking about this, everything from the enablers on the research side to what we can do in terms of clinical implementation. So I'm not going to go into this in much detail because that's exactly what this course is about, trying to pick this apart stage by stage and understanding what are those things that we can start building together, where we are now and where we'd like to be going. So there's certainly lots of challenges ahead. One of the great challenges is that in New York, I love the idea that we have everyone from every part of the world. From a genetic point of view, that's really quite challenging because we have genomes from all four corners of the world. But if we don't start understanding what those normal variations are and those abnormal variations, we will have genetic haves and have-nots because there will be certain minorities that we simply will not be able to give an accurate interpretation of their genome. And so partnerships that we have with our local communities, be they from the Middle East, be they East Asia, be they African American, we need to build stronger partnerships, and we actually are strategically able to make great inroads because we do have such diversity right here in our backyard. As we've talked about many of these other things, there are challenges. There are challenges in cost, there are challenges in demonstrating clinical utility, there are challenges in time, there are challenges in education, both of patients as well as providers. And increasingly, and those of you who are reading this and hearing about it in the news this week or very recently, there are challenges from government regulation in the FDA, and increasingly whether or not they're going to regulate this whole industry the way they have 23andMe, or whether or not there should be other police out there like CLIA to be able to do this. So this is going to be important as we go forward and do this. So I just want to say that for the things that I've talked about, clearly this is team science, and this is team science in a big way. I think it's a lot of fun to do that way, but it poses its own challenges in terms of communication and working together, but those are the opportunities that we have. So hopefully this gets you energized about why you're going to bother spending late Thursday to be able to come to the course and think about these things. But as I said, what we're going to do now is break these into now bite-sized pieces to do deep dives in lots of the things that we've started talking about, and hopefully both pick your brain to be able to think about solutions to this, as well as I hope to start forming communities of people who are going to start to tackle these types of problems.